good evening, depending where you are. And thank you for joining again the African Seminar Series. Today, we'll be glad to have Dr. Leda Mostaso-Guidolin for joining us. Leda is currently an assistant professor at Caltech University in Canada, where she was a medical physicist by training. She then obtained her PhD from the University of Manitoba in Canada, where she developed her expertise in imaging analysis method to characterize atherosclerosis based on multimodal nonlinear optical microscopy. And after completing her PhD, she undertook her postdoctoral training at University of British Columbia at the Center for Heart and Lung Innovation, where she then worked on the application of imaging modality in respiratory research and development of 3D in vitro models. Currently, her work focuses on combining microscopy and tissue engineering to characterize biological tissues and to better understand the changes associated with fibrosis and extracellular matrix remodeling in diseases. Without further ado, Dr. Mostaso-Guidolin, please take it away.
Thank you very much for your kind introduction. So hello, everyone. Good morning, good afternoon. Let me just share my screen here. I assume you all can see it. Perfect. Okay, so it's a pleasure to be here today, meeting this amazing group. I have tried to join some presentations in the previous month, and it has been great. So I hope you enjoyed this talk today. I'll be talking a little bit about bioprinting, but I will show you mostly a very interesting story in terms of how can we make use of the amazing images that we can get with imaging modalities, and then of course, use that to our advantage to address some questions that we have in biology and then discover new things, right? So today's story is going to be interesting in the sense that I will be talking about in the domain of several disciplines. So I'm going to talk a little bit about cellular biology, biochemistry, tissue engineering, microscopy, of course. And to illustrate that, the story that I have today, I'm going to tell you all about how we took advantage of using my optical microscopy to drive some information from the part of characterizing tissues, sections from human tissue that we had, all the way now to the current development of 3D bioprinted models. So I would like to start asking a very simple, but at the same time interesting question, which is what holds us together? So as animals, as humans, we know that we have different types of tissues, we have different types of cells in our bodies, they all do different things, they have different functions, but there is one component in there that helps these cells to communicate, to move around, and to form the structures that we need to have all these functions performed. And this structure is called the extracellular matrix. So that's a network of proteins, just like the figure on the left. I hope you can see my cursor here. But so that's a network of different proteins that they are all, they form this mesh in there, and the cells are able to navigate, and they also can get some biochemical cues from the extracellular matrix. It's very important when we are talking about aging or the repair of wounds. And that's one of the main subjects of my research. So I'm very interested in characterizing what's going on with the extracellular matrix per se, and what's the effect that it has on the cells. So here we have a very oversimplified diagram of how this matrix looks like. So the cells, which is represented here with these yellow bars, they are attached, of course, to this matrix. They can use that as kind of a highway for them to move around, to communicate. And of course, as I mentioned, we have different types of proteins and other components in there. And one of the main ones that is responsible for making up a lot of this structure of the matrix is what we call collagen fibers. And that has been one of the main proteins that I have been interested in characterizing and looking how it's changing depending on the disease or depending on different factors, and how that is affecting the cellular communication, for example, or differentiation and so on. So why exactly am I so interested in collagen? So collagen is one of the hallmarks of animals. We, for example, as humans, we have over 80% of the proteins now in our body, it is collagen. We have different types of collagen in there. So it's a very important structural protein. And what makes it very special is the fact that the collagen fibers or the collagen proteins, they have a very unique structure. So as you can see here in this diagram, the fibers, they are made of a very interesting structure that is composed by fibrils, which are tiny, tiny fibers inside of the main fiber. And then these fibrils, they are made of triple helix structures. Just like the DNA that we have that nice structure of a triple helix, inside of the collagen fibers, fibrillar collagens, we have a similar structure. Imagine that you have a very thick rope, and then you have all those threads in there. So that's more or less the idea of a collagen fiber. So that's why we have such a nice tensile strength. It's similar to a steel cable, for example, per se, and it's all thanks to this amazing and unique structure that we have in this fiber. So no wonder that's one of the main proteins that we have in our body, making up for our structure that we need and all the support that we need, right? However, if we look down at different abnormalities or different disease and pathologies that we have, if you dig enough, you will notice that most of them will have some sort of a collagen-related disruption, some collagen-related abnormality happening. For example, if you look at skin disorders, gum disorders, even atherosclerosis, for example, you will have a little cap of collagen being formed on top of these lesions. Lots of respiratory diseases will have accumulation of collagen that will prevent the lungs to function properly. So collagen is a very interesting candidate for us to look at if we want to understand how the microenvironment of these cells is changing and what the effect it's causing in different applications, even cancers. Sometimes the microenvironment that we have in the tumors, we do notice an accumulation of collagen. So it's a very important aspect for us to take a look. And here I just have an example. All of those images here, they are from fibrillar collagen. And what's interesting here, although you're seeing they are very different in terms of structure, they all came from arteries and from atherosclerotic lesions. So it's very interesting for us to see from the point of view of structure that this collagen network or the orientation of these fibers is changing a lot, even though we are looking at the same, let's say, the same pathological condition, the same organ, the same tissue, we have a lot of variability in there. So one of my research interests relies on how can we take advantage of all these changes that we are seeing? How can we actually quantify these changes or have some objective ways to measure everything that we can observe? And today I will share with you a little story. The background of this story is about asthma. So I'm going to tell you how I took advantage of using optical microscopy to characterize changes that were happening inside of airways. And then I will finish the talk mentioning the current work related to actually bioprinting. And yeah, so asthma is a word that comes from the Greek, and it signifies short of breath. So I'm sure probably all of you probably know someone, or maybe you have asthma, and you notice that when you have asthma, you have a little bit of a difficult breathing. And that's because your airways are a little bit obstructed. So here is an example of the first image that Dr. Henry Salter managed to get of the lung. As you can see, the image, of course, is not, we cannot see much details in there, but that was the first evidence of that we had of the airways. So you can see here those little two holes in here in this tissue, they were airways, and that marks the beginning of studying, of someone attempting to study what was happening inside of the lungs and trying to address what was the issue for people who had respiratory diseases. 65 years later, of course, we came a long way. And Dr. Claude Masson developed a different type of histological staining. And that was a very groundbreaking research because at that time, finally, we managed to have a way to differentiate, even though a little bit, different types of tissues inside of, for example, in this case, the airways. Of course, that was used to characterize different types of tissues, but here we have an example of an airway that's a cross-section of the airway stained with this Masson trichrome stain. So some of you probably are familiar with this, but in case you are not, everything that's kind of bluish, greenish in here is collagen. The purple reddish structure around here and here is the smooth muscle cell. And then here we have the epithelial cells. You can see a little bit of the cilia. Here in the middle, we see mucus. And when you compare the structure from someone who has asthma with someone who has a normal lung, you can see the difference is striking. And so that was a very nice breakthrough in terms of imaging, right? And then we came to 2021. And more than 90 years later, that has been the way we have been analyzing and looking at lungs and trying to address, understand the issues that are happening in the airways and trying to understand what's going on with different diseases. And that was very interesting because as a microscopist myself, I was very interested in bringing new tools to this area. So I started looking into how can we take advantage of the new microscopy tools and some not so new to actually see the structures that we have in terms of the collagen fiber. When we look at this image, it's very clear that we can see, for example, the amount of collagen, again, the blue greenish part. We can see that we have more accumulated in here compared to the other side, but we cannot really see the fibrillar structure. We cannot see the individual fibers in there. And that could be something interesting, right? So if we are trying to understand how the extracellular matrix is changing, how the network in this matrix is changing, we need to have a way to really assess how the individual fibers are being modified depending on the disease in this case. And one interesting fact about asthma is that asthma is associated with chronic inflammation. So there is what's called a fibrosis. So fibrosis is accumulation of collagen. As you can see, as I mentioned to you, all this amount of collagen that we have accumulated inside of the asthmatic airways is actually fibrosis per se. So we have more collagen than we should. Therefore, the airways becomes a little bit obstructed. The air cannot flow in and out. And that's the reason we have that shortness of breath. And there is no cure. And of course, that's clear because we don't understand exactly what's going on in this mechanism. We don't know exactly what's causing this fibrosis. What are the cellular mechanisms behind it and why this collagen is being accumulated in such a huge amounts in there. And that was the starting point of one of the research lines that I have been carrying on. And as I mentioned before, so currently we can visualize collagen and elastin, which are some of the main components of the extracellular matrix. We usually do it using immunohistochemical staining. Here are some examples again. So we have the Masson trichrome, which is the one I showed. We have some other options in there if we want to visualize some structures. But again, if you have done a little bit of work with staining, you know, it's time consuming. We have to follow a very long protocol sometimes. It is kind of low, it has a low spatial resolution in terms of assessing the individual fibers, and it will require the use of several chemicals in their additional stains for us to manage to get the pictures like this. And one alternative to using stains relies on what's called the nonlinear optical microscopy. So it's sometimes it's known as multiphoton microscopy. If you come across with these names, those are a family of different techniques that we can use and acquire signal or generate images based on the properties of the material that you are trying to image or the properties of the tissue. So you don't have to stain, you don't have to add anything in your tissue in order to get some images from, let's say, the biomaterials that you want to image. Here I have some examples to show you how we can compare what kind of information we can get when we use the nonlinear optical microscope. So for example, here we have some regular stained tissue sections. We used the different stains I was mentioning. So we can see, for example, everything that's kind of blackish, darkish here is the elastin fibers. The pinkish red is, among other things, a little bit of collagen. And then using two different types of nonlinear optical microscopy, we can actually see collagen fibers, as shown here in this first pair of images. Then we can also take a look at the elastic fibers in there. And here is just a combination of those two images. So we can overlay them and see how the collagen fibers, for example, are distributed in the tissue. And what's amazing is you can probably notice that now we finally have some resolution to actually see structures or fragments of collagen in there. And that's all done without the need of any stains. So that's a very cool technique for us trying to understand biological materials, right? Because we don't have to disrupt the environment. And at the same time, we can get very interesting images that will allow us to understand a little bit more what's going on in terms of the biological mechanisms in there. So just very briefly, an overview of how this nonlinear optical microscopy works. So in general, in traditional microscopes, we will have just let's say a white light source. These will be emitting multiple wavelengths, so different colors. They are diverged, they are dispersed, and usually we need staining to actually see the tissue. When we use a nonlinear microscope, for example, we are going to rely on lasers. So the laser light is coherent, meaning that we have all the color is the same that's going to be emitted from this laser. We can have decollimated, so meaning that we can have a very well focused light beam hitting the sample. And the advantage is that we can generate some specific physical phenomenon using laser light that allow us to visualize these biological features without requiring any staining, any preparation. And since we are talking about collagen today, I'm going to just explain to you again briefly how second harmonic generation works. It's one modality under the umbrella of nonlinear optical microscopy. Under nonlinear microscopy, you have different types of modalities. One of them is second harmonic. And the way it works, it's a very good modality for visualizing fibrillar collagen. And why is that? The way it works is we need to have what's called the optical nonlinear medium. And then you have your laser light that will have a specific frequency. This laser light will interact with this special nonlinear medium, and that's going to generate what's called a second harmonic wave. So it's a wave that will have 2 times the original frequency of your laser light. And you might be wondering, okay, so how do I find this special nonlinear medium? If you remember a few slides ago, I told you that collagen fibers, they have that unique structure, right? So they have that triple helix, they have the fiber made of fibrils, and then we have all that triple helix making the fibrils. So that's exactly what we need in order to be able to generate the second harmonic wave. So the lack of centrosymmetry that we have in collagen fibrils or fibers, it's what's responsible for allowing us to actually generate this wave. And that's the signal that we detect when we are dealing with second harmonic generation. So the idea is we have our biological sample in here. All the collagen fibers will generate this wave, and that's why we can actually get amazing images like this one without the need of any staining. So that will come thanks to this unique property of the collagen fibers. And so going back to the biology question is, so I started wondering, okay, so we have this amazing microscopy technique that can visualize fibrillar collagen without staining. We have been relying on staining for a long time to try to understand the changes in asthmatic lungs. So what are the structural properties of the collagen in the remodeled airways, right? So now we can see individual collagen fibers. So what's going on in there? How much are they changing? We know that we have more collagen in asthma, but at the fiber level, is it changing significantly? So that was one of the research that I have been carrying on for a little while now. And one of the hypotheses that we had was, okay, so maybe the fibrillar collagen is more fragmented because of we have a defect in the with the cells that make collagens in asthma patients. So we started trying to address this hypothesis through the years, and that's when different ways of testing this hypothesis came along. So we started looking at tissue sections, then we went to 2D models, and then now I'm actually working on 3D models for trying still to address and to validate all the information that we have been gathering. So this morning, or this afternoon, I'm going to show you some of the results and some of the interesting findings that we got from the different types of models per se that we had. So all this work, again, it started with tissue sections, then we moved to 2D cultures, and then now we are currently looking at 3D bioprinted models. So the idea is we are now combining nonlinear optical microscopy, which is a label-free technique, and now we are actually developing or taking advantage of this information that we have in there, and developing some image-based, calculating some image-based morphological metrics to characterize the fibers and also take a look at the cells. So the big question is, are the asthmatic fibroblasts able to remodel fibrillar collagen? In case you are not very familiar with this biology, fibroblasts are the cells responsible for making fibrillar collagen. So we want to know if the cells, they are working properly to make these nice fibers or not. And one important aspect that we had to address was related, okay, so now we have the samples, we have a very interesting microscopy technique to look at the collagen fibers, but how do we make sense of these images? How can we quantify the information? Or how can we extract the fiber structure from these images? Usually the way we deal with image analysis is we have an image. I'm sure all of you know this very well. So an image is made of pixels. The pixels, they do have different intensities, right? Which is related to the amount of signal that you are collecting from your sample. And therefore, if we do what's called the first order analysis, or if we rely on first order statistics, we can kind of have a correlation with, let's say, the amount of collagen, the amount of elastin that we have in the sample. So we can do simple math with this number, these intensity numbers, and usually we can get some idea of what's going on in our images. However, sometimes we might face interesting problems like this one. So you can see in this example that the three images, they are completely different, right? But half of them have white pixels, half of them, 50% of the image has white pixels, 50% of the image has black pixels. And if we calculate, for example, the average or the mean, it's going to be the same number. It doesn't matter that they are distributed all in a different way. So how can we distinguish those images? One way to address this is by calculating what's called the gray level co-occurrence matrix. I won't go into too many details today, but the idea is we are going to look at the distribution of intensities in an image. We are going to look at what's the relationship between one pixel and its neighbor. And we are going to quantify, for example, how many black pixels have a neighbor that's also black in diagonal, horizontal, vertical, and so on. How many white pixels have a neighbor that's a certain shade of gray, and so on and so forth. So by considering these relationships, we can track structural changes happening in the image. So in our case, we can actually see the fiber morphology. And there are several parameters that we can calculate based on this matrix. One of them that's very easy to visualize is called entropy, just like in physics. Entropy is a measure of the lack of organization. So the more disorganized something is, the higher the entropy. And for example, if we look at those three images that I just showed you, and we calculate the entropy, for example, that's going to allow us to distinguish the structure that we had there. Okay, so going back to the original problem, based on texture analysis, I started looking at all the images that I had from collagen and elastic fibers, and I started calculating different metrics to see if we could somehow obtain a parameter that would tell us what was changing inside of these airways when we compare normal people with asthmatic donors. And it was very interesting that the entropy, for example, we noticed that in asthmatics, the collagen fibers were actually more disorganized when compared to normal. The intensity, of course, was higher, but that we knew from the staining. We could see that we had more collagen there. But this piece of information here was a new finding. So we finally were able to see that the collagen fibers were more fragmented. They were not so organized when we compared to normals. And that started to lead us to a whole different set of analysis, because another aspect that we could look at was related to collagen orientation, for example. Here we can see in normal airways, we have the collagen fibers well aligned, well organized. When we look at the asthmatic, we can see like all these fragments, they are all a little more not evenly distributed. That's another metrics that we could extract from these images. And of course, since now we can see individual fibers, we could assess some characteristics related to the fiber morphology. So for example, we could look at how thick those fibers were. We could look how far away they were from each other, comparing asthma and controls. And we noticed that the fiber spacing, the distance between them in the matrix was actually different. So in the asthma case, we had a larger spacing when compared to the controls. The thickness did not change, but this spacing did change. So up to now, we know that nonlinear is very good for visualizing fibrillar collagen. So we saw that the fibrillar collagen is disorganized in asthmatic airways. The elastin, we did not find any significant difference, which was very interesting. And at that point, we decided, okay, so now we have to understand the cells and the mechanisms responsible for this disorganized extracellular matrix environment, right? So what's driving all these changes that we noticed in tissue? And so again, just quickly, so as I mentioned before, the fibroblasts, they are oversimplifying things, the extracellular matrix builders. So in this case here, we have a fibroblast cell. When they undergo some activation, that can be physical, that can be chemical, they will start producing collagen fibers. So we wanted to understand if in asthma, the fibroblasts had any defects or they were having any issues making the collagen fibers, right? Because we noticed that it was more disorganized, the matrix was more fragmented, and but why was that happening? Was it the cells that were not able to make the matrix properly? So are the fibroblasts producing this fibrillar collagen or not in asthma? What's driving these changes, right? So that was the second step in this very long-term study. And that we started looking into 2D models, 2D cell cultures. So we developed several in vitro assays for collagen remodeling. So the idea was we could put some collagen that was not in a fibrillar form, in a gel, let's say, seed some fibroblasts in there, wait a certain time, and if they are working properly, they would be able to remodel this collagen, make the nice fibers, and everything was going to be fine. If these cells, for some reason, had some defects, it was very likely that we wouldn't see the collagen fibers being developed as the way they should. So that's exactly what we did. So those are pictures of showing some examples of these in vitro assays for the collagen remodeling. They are probably a little hard to see, but if you follow more or less this line, you might see the collagen gel, as we say. They were seeded with fibroblasts. We waited 72 hours. And then we looked at how much of fibrillar collagen these cells, comparing asthma and normal, were able to remodel. And here is again from microscopy, a very nice picture of 2 fibroblasts and all the collagen blue being produced by these cells. So we tested some of the mechanical properties of these gels because that was going to give us some indication of how good the fibers were. Remember, collagen gives us structure. So if we have nice, strong fibers, the gel was going to have different mechanical properties. And what we found out was actually that the asthmatic fibroblasts, they could not make fibrillar collagen. So when we didn't have any cells, we we had a control there, so we didn't add any cells to the gel. So for us to have a reference, when we see that the healthy fibroblasts from normal, from controls, we noticed that the gel was more contracted, meaning that when the cells start to remodel this collagen, they start pulling the pieces of collagen of the gel, and then they make the nice fibers, therefore the gel is contracted. In the case of the asthmatic, you can see from this picture, the gel is a little more spread, meaning that the cells were not able to make these fibers. We looked at those gels using, again, second harmonic microscopy, so one type of nonlinear microscopy. And we could indeed see that, for example, when we looked at the control gels, we saw a lot of fibrillar collagen. When we looked at the asthmatic, the signal was a little more sparse, meaning that we did not have as many fibers as we had in the normal case. Again, we calculated those parameters from the images, so the intensity, it dropped when we looked at the asthmatic, so meaning we have less fibers. The entropy was, again, higher for asthma, meaning that the arrangement of whatever little bit of fibers they made was more disorganized than it should be. And that led us to a mini conclusion saying that, okay, so the asthma-derived fibroblasts, they are indeed defective at collagen 1 contraction. They cannot make these fibers. But then we didn't stop. So we started wondering, okay, so now we know they have this defect, but what's driving these cells to not be able to remodel the collagen? So we started wondering if the cytoskeleton of these asthma fibroblasts were defective. What kind of difference they would have that it's not allowing them to actually remodel the collagen properly, right? So we looked at different types of proteins and gene expressions related to cell movement, cell contraction, cell repair. So here is a part of the list that we looked at. And surprisingly, we did not find any difference when we looked at asthmatic and normal cells. We were expecting to see something, at least to explain why these cells were not able to make the matrix properly, but the story became only more interesting. We tested some genes, some proteins, expressions, and so on. Nothing was different. It was very puzzling at that time. And eventually, we had to have this mini conclusion number 2, which told us that the asthma fibroblasts, they did have a normal functional cytoskeleton. Again, we were not expecting that. That was a very big question mark for a long time. And eventually, after a lot of research, a lot of reading, a lot of talking with colleagues and potential collaborators, we decided to test one last suspect. So we decided to explore and test if decorin was changing comparing those two cells. So when we look at the collagen fiber, we have these little proteins, little structures in here, protein-like, hanging in there, which is the decorin. So the name came from it decorates the collagen fiber, and it's very important. It plays a very important role in the matrix assembly. So when we look at one of the functions of decorin, that's the one that's going to allow the fibrils inside of the fiber to be properly spaced. And without keeping the suspense much longer, so after running some tests, we did find that the asthmatic fibroblasts, they had lower expression of decorin, meaning that they were not able to properly space the fibrils inside of the collagen fiber. So that was probably the cause that was not allowing these cells to actually make the nice long collagen fibers as we were expecting. They just could not assemble them properly. And so that was a very interesting and long story. I'm just summarizing the main bits for you today. But then when we got this response, we decided, okay, so now let's just take a quick look at the fibrils, because we looked at the fibers using nonlinear optical microscopy. We now have evidence that the decorin has different expression when we compare normal and asthmatic fibroblasts. Let's look if indeed the fibrils, they are not properly organized, right? Because that's what one should expect. And that was exactly what we found. So we went on to use some transmission electron microscopy to look inside of the collagen fibers. Here is a cross-section of a collagen fiber. And as you can see, the difference is striking when we compare controls and asthmatics. So in the controls, we have all these fibrils very well organized, very well aligned. But when we look at the asthmatic, we have all this disorganized structure in there. So that's telling us that, yes, there is no way the fibers were going to be properly assembled, right? So the problem or the differences in structure is starting inside of these fibers. And so the conclusion of this study was that, yes, the asthma-derived fibroblasts, they were defective at the packing the fibrillar collagen 1 because of the lower decorin expression. And that was all very interesting, very nice. It was a very nice example of how microscopy helped us to go on and have these conclusions. But we know well that tissues and parts of the body and so on, it's way more complex than that. So we have different shapes, we have different information that we have to account for. And that's where I am right now. So now I'm moving from the 2D cell culture into the 3D bioprinting world. So in 2D, you probably know well, we have usually Petri dishes, any kind of recipient where we seed the cells, we let them grow, and then we track what they are doing. But there are some limitations to these models. We have some issues in there related to how the cells will behave. There might be some significant differences that we shouldn't be seeing just because of the way the cells are able to move around. When we move to 3D bioprinted constructs, we allow the cells to have this freedom and move around in a little more realistic environment per se, and therefore hopefully express the proteins and genes the way they would in real tissue. And so now I'm working mostly trying to understand fibrosis in general, meaning the accumulation of collagen in different tissues, in different organs, not necessarily related to lung tissue or asthma, but I'm very interested now in looking at how can we create 3D models using bioprinting that will allow the cells to behave exactly the way they should in real tissue, and therefore study how the microenvironment, how the extracellular matrix, how the collagen changes might drive differences in terms of the cellular communication, the cellular behavior, and so on. And yeah, so I mentioned, I'm just finishing up, but I just mentioned quickly before. So fibroblast cells, they are present in different tissues, not only in lungs. We have them in skin, we have them in different parts of the body. They do suffer some stimuli they need to suffer some stimuli in order to differentiate. That can be mechanically or chemically again. So for example, when you cut your skin, the wound healing process will start. Oversimplifying things, that's a mechanical / chemical stimuli that will ask the fibroblast to come around, make more collagen, and therefore close the wound. Again, there are lots of inflammatory markers and so on, but in a nutshell, that would be one aspect we could look at. And so in terms of fibrosis and all of these collagen remodeling, there is $1 million question open, which is the fibrosis causing the inflammation that we notice in the tissues, or is the inflammation causing the fibrosis? So are we injuring the tissue and then the cells, let's say, quote unquote, go crazy and try to address the changes or the injuries? Or do we have the inflammation happening first and then that's what's triggering the cell? So that's one question that's limiting the development of several therapies, because we don't understand exactly what we should address. We should address the amount of collagen, the collagen remodeling, or we should address the inflammation response, right? And which one should be the therapeutic target? So the fibrosis or the inflammation? And the steps are, of course, not well understood yet. So we have to still do a lot of work to understand the molecular mechanisms that the differentiation of the fibroblasts occur. We don't know exactly why that occurs. And we know that the extracellular matrix has an effect in there, but it has not yet been well characterized. And another question that's open is how the cellular communication is affected when we have an abnormal mechanical environment. As you saw before, when we have a lot of collagen in fibrosis and so on, the cells don't have their environment the way it should. Therefore, that's affecting their response and how exactly that's affecting, right? So 3D bioprinted models can help us with that. This is an area that's not very old, so there are lots of new research coming up from this. And one of the questions that I am directly involved with, I'm trying to create a matrix that of course is synthetic, but it would allow the cells to behave as close as possible to the behavior that would observe in vivo tissue. So here's an example of a bioprinter that I have in my lab. So as you can see, we can put in here what's called a bioink. So the bioink is basically anything you would like to use as a matrix. So that can be collagen, that can be alginate, that can be a combination of proteins. It can have cells, not have cells. And then we can use some softwares like CAD that engineers, architects use to develop houses and so on. We can use something similar to that to build constructs and define different structures. And we can build, basically we can print, bioprint anything we want. We can play with the physical parameters, how stiff the material is, how soft, how many cell types we want in there, how many proteins and so on. So as you can imagine, it's a very interesting field, but also very challenging. And here I just have some examples of a work that we have going on right now, just testing different types of bioink. So by changing the proportion of collagen and fibronectin, for example, we can see that the cells, the blue is collagen, green are the cells. So you can see the cell shape is changing quite dramatically depending on the composition of this oversimplified matrix. So that's something that we have to address as well. So how can we make the cells happy in this environment? And what exactly are the ingredients that we have to put in these bioinks for them to behave the way they should? And that's an ongoing work. So we are exploring the effects of bioinks on fibroblasts. So for example, you can see from the pictures very clearly that the samples look completely different depending on the proportion of these proteins. And the cells also look completely different. Some are very unhappy, some are doing okay. And one thing that we have noticed so far, for example, is that if we don't add any fibronectin, we allow the cells to remodel thicker collagen fibers, but also they become more fragmented. So that's all ongoing. I don't have all the answers for you today. And here is just one last image for you. We are now also going back to the asthma problem. We are now printing hollow tubes structure. So we have different components in here. The idea is to have air flowing inside of these tubes to simulate an airway structure. So the idea is to have, of course, the bioink that's going to mimic the proteins that we have in an airway wall. We will have different types of cells. In the beginning, we are going to start with, let's say, epithelial cells inside, fibroblasts outside, and then see how they behave in this structure. So, and here is a picture, again, from microscopy, showing this is the hollow tube. Sorry. So here is the hollow tube. We can see a little bit of the matrix being developed, a lot of cells distributed all over the place. We can add inflammatory markers in there if we wish. We can use those constructs once they are established to test different therapies. And again, going back to second harmonic microscopy, we can see how the collagen structure is changing over time. And so we are now in this road of trying to understand exactly how we can translate the normal behavior of a cell into an artificial structure. So moving forward, those are my plans. I am still working on characterizing fibroblasts, but now I'm looking at 3D bioprinted matrices. And the final goal is to develop some lung niche models, meaning that we can have an artificial lung, and so we could speed up the testing of therapeutics strategies. And then, of course, track how these medications or therapies are affecting the cellular behavior, right? So that's always the holy grail of almost any biomedical research. And I just would like to finish thanking a huge group of collaborators and coworkers that have been made all this work possible up to now. And if you have any questions, if you would like to connect later on, please feel free to send me a message. I'm available, so it would be a pleasure to connect with you again.